We are taking our very last topic for today. And so after today, I will say that it's enough for the semester. And you will just have to use all the materials that is given to you, the presentation slides, the videos, the book chapters, and the self-assessment questions to start preparing for your exams. So in other words, today is the last lecture that we are having for the semester. And we will finish up with what we started last week with biotransformation. Last week, we took pharmacokinetics. And so today, we will continue with pharmacodynamics. We did talk about the fact that pharmacokinetics dealt with what the body does with the chemical substances that get into the body, looking at processes of absorption, distribution, metabolism, and excretion. And pharmacodynamics today, we're looking at those response relationships of the chemical substances that we are exposed to. We'll talk briefly about drug-receptor interactions. We've actually done a lot about ligand receptor interactions. It's just the same thing. In this case, the drug is the ligand, and then to be interacting with the receptors. We did talk about receptors when we were looking at the endocrine system, the hormones, and we were looking at the mechanisms of action of these hormones. So it's the same way that you have these biomolecules such as the hormones, or say maybe enzymes, acting in the body. Is in a similar way that you have drug substances, which are, say, analogs of these biomolecules, so that when they enter into the body, they are mimicking one of these biomolecules. So when we look at mechanism of action of these substances, I think you'll get it much clearer. Therapeutic and toxic effects too, we did talk about it when we were doing the pharmacokinetics. We did see the kinetic curve, and we referred to the safety corridor, the maximum concentration, which is a threshold above which you start to have toxic effects, minimum therapeutic effect concentration, and the distance between them being the safety corridor. So therapeutic toxic effects really there's not much to say about it. But as we go along, maybe when we are discussing some of these things, things of that sort may come up. But much of our focus is going to be on the mechanism of action of drug substances. So, I mean, not to make it boring, take some specific examples and take a look at the mechanism of action of these drugs. So that's our plan for today. And by the end of maybe 1 hour, 30 minutes thereabouts, we should finish going over it. And if you have any comments, any contributions, anything you want to share with us, any questions, you will have the floor to speak, or you can write in the chat as the lecture goes on. You can write in the chat and I'll respond to them. Or maybe at the end, you can write your questions there. Or the microphone is available. And once you raise your hand, I will enable your microphone. You unmute yourself and then you speak. All right, so we start with the dose response relationship. Now, the dose-response relationship which we are looking at here is that, referring to lethality of a substance in relation to its dose. You can look at it as say therapeutic effect. Say in place of lethality, you can put therapeutic effect there. It will give you the same idea of what we are looking at. But this is a comparison of three substances: substance A, substance B, and substance C. And what you notice is that, so on the lethality axis, you have from 0 up to 100. And what you notice with this dose-response relationship is that the relationship is non-linear. You don't see that it's just a straight line to say that once the dose increases, you have the effect, the lethality also increasing proportionally in that manner. So generally, you have these dose-response relationships being some sigmoid curve, as we are seeing, or maybe some parabolic curve. You see things like that. But an interesting thing that you should pay attention to is that you see that the dose regimes, the dosage regimes are divided into, say, different zones. Let's say 4 different zones: zone 1, zone 2, zone 3, and zone 4. Now, if you pay attention, you realize that if you take zone 1, you see that the lethality of the substance A is more than that of substance B, and that is also more than substance C, which means, at the same dosage range, the effect of substance A will be higher, or is the highest, followed by the effect of substance B, followed by the effect of substance C. Then when you come to the second region, region 2, you realize that that is not the case as you had in the first dosage range. That region 1, you see that the effect of the substances are not the same. You rather see that the effect of substance B becomes more than the effect of substance A, and then followed by substance C. Now, clearly, what this is telling you, when you continue, you see that this continues to change. Like if you go to the range 3, here too has changed, so that now you see that the lowest effect you are having is from substance A, and it is substance B that is giving you the highest. And by the time you get to the end, which is the 4th region, you see that substance C has the highest effect, followed by substance B, and then the lowest is substance A. Now, what this means is that you will not be able to determine accurately the effect of substances when you are comparing different substances, their effects, if you are just looking at one particular region. You have to study the full spectrum of these substances to be able to tell, how these substances are behaving in different dosage regimes when you are comparing them. And so you may not be able to tell if perhaps you are just looking at the dosage range of, let's say that you are having a concentration of the substances. Say the first here you mark it as let's say that is 0.1 moles per mil, for instance. That is to say concentration, and you continue 0.2 mils. And then maybe this is 0.25 mils. And the last end there, like say maybe 0.4 mils, for instance, 4 mils per liter, or say in molar, all of them molar. Now, it means that if you just take one concentration range and you are comparing the substances, you will be making an error to just say that A is the most effective than B, and C is the less effective, and then you remain there. But if you had continued to look at different concentration ranges and compare the effect of these substances, you would have come to the conclusion that actually the effect of the substances vary in different regions. So there is this dose response relationship with substances when you are comparing substances to choose. I mean, in researches of let's say drug development, drug research, it is important to check different regions in comparing these effects. Let's say in terms of medical substances, pharmaceutical substances. That is to say, you are comparing therapeutic effects, or if you are mostly studying the toxic effects of the substances, then you have to be looking at different concentration ranges in order to be able to determine these relationships of these substances pertaining to the dosages that are being administered. Now, for every substance, there are some specific properties that are also of interest. I mean, first of all, for every substance that is being administered, there is what we refer to as a background effect, which really below that background effect, there is no therapeutic effect. That is to say, if a substance is taken in within a certain very low concentration, that concentration does not have any effect at all, except that yes, even if it is that maybe you are measuring with some instruments, there will be some background noise. It's the same thing you do with your equipment like photospectrophotometer or some HPLC or anything. You first do like some blanking. You do measure blanks of solutions so that you take that into consideration. You subtract that from maybe if you are measuring absorbance or anything. I mean, it's a similar thing over here. You are seeing that there is some background effect that if you have concentrations of substances at a very, very low concentrations, you really don't have any significant effect occurring. So that's a background effect. And then you begin to see, so the curve, those curves that we saw, you can see clearly that is the same, similar curve that we are seeing, sort of a sigmoid curve. There is that rise in the effect up to a certain point, and then that then plateaus and becomes constant, because at that point, it's like you have a saturation of the substance. So there is really no difference again. That's the highest, whether it's a therapeutic effect or it's a toxic effect. That is the highest of the effect that you can have with such substance. Now, so that's curve 1 that we have looked at. But there are some interesting things over here. We can see there's some curve 2, curve 3, curve 4, which are, just showing within the region where you have a very, let's say, a super minute concentration, because here you can see clearly that the curve is even starting after that. So in this region where you have such a very low concentration of the substance that is having no effect at all, or let's say it's not even recognizable uh, in that sense. The concentration is so low. We say maybe a super minute um, amount of the concentration. Now, this is also taken into consideration, particularly when studying toxicity of substances, because what happens is that in some, populations, even though this super minute concentration, particularly with things like radiation, you can have um some disproportionate effects among the population. Some people will experience certain effects, uh, which really is not that significant for some, but for some, that can be also a little bit, uh, significant, because as you can see, the curve goes a little bit above the background effect. If you look at curve 2, you see that actually you have that effect being higher. So it can be high in some people up there, and in others it can be so low and just at a very you know low concentration, where you see the intersection between the dash lines and the curve number 4. That is the only little effect that will be felt in such persons. But in all of this, mostly when this uh, super minute concentration effect is being studied, it is the curve 3, which is a straight line. I mean, it's just an estimation of what can happen among some populations with such very minute dosages. So it is much more convenient to just use a linear relationship than to now be using these, curves, which are actually, like, this is actually, say, maybe what you observe, say, curve 2 or curve 4, but you usually will use a curve 3 just to estimate the effect that such superminute concentrations may have in some persons. So in that case, you can see that we are having this region, PT, labeled PT. You have that concentration there labeled PT. You have another concentration here labeled PT, what we are referring to as a practical threshold. And the practical threshold is the lowest concentration or the lowest dosage of a substance from which you begin to experience a therapeutic effects, if it's a pharmaceutical substance, or if it's a toxic substance. You'll begin to experience significant toxic effects above the background effect. So if you are looking at the super minute concentrations, then that intersection there for these curves is what we are referring to as the practical threshold. That concentration there is a practical threshold. There are some two other things that are here, LD50, and that's to say lethal dose 50. You'll see this also when you take the leaflet in the medicines you buy. You will see when some pharmacodynamic studies, the results are written. You can see something like LD50 over there, which is to say that the lethal dosage, that is the dose that when it was given to say experimental animals, when it got to that dose, 50% of the the laboratory or the animal models which were used to study the toxicity of the drug substance died at this concentration, the lethal dose, 50. So 50 means 50%. 50% of the animal models used to study the toxicity will die at that dosage. Sometimes you see it written as LC50, which is a lethal concentration, 50% of the population that die at that. So usually the dosage regimes are set, below these concentrations. You shouldn't you come anywhere close to this if it's a toxicity. Shouldn't even come close to any, because if you are, of course, if it's a drug, the drug is supposed to rather provide a therapeutic effect and not a lethal effect. So you have to understand that any substance that you are taking in, it depends on the dosage that is being taken. That substance can either be therapeutic or it can become toxic. So the same substance you are taking, which is having a therapeutic effect at a certain dosage or certain concentration, that same substance, when it is within a certain concentration range, that's why we talk about overdosage. That can then have a toxic effect. And so you see, then from here, you have that gradual increase in the effects all the way until it gets to 100% of the effect of the substance. All right. And now, another thing that I would like us to talk about is when you take, let's say, two different substances. So we have substance A. So look at the vertical axis, which is on the left-hand side, 100% A. So, this is A, substance A, and then there's substance B on the right-hand side. So that horizontal axis is referring to amount of substance. And then the vertical axis is showing the effect that the substances will be having. Now, what we are showing over here is the three different things that can happen when you combine two different substances together. This is to say, like drug-drug interaction, how maybe two drugs interact or two or three drugs interact. We have what we refer to as a summation or additivity, summation effect or additivity. That's to say that when you have two substances put together, both of them will have a summative effect or an additive effect, like a combination of the effects that they are having. That is represented with the curve 1. So you have that effect is increasing, gradually, sort of say linearly, proportionally that way. Now, there's another thing which happens, which is referred to as potentiation, also referred to as synergism. A lot of times, I notice many people make that mistake. I think among you, even the students, when I sit on your panels and you are defending your project works, a lot of you, when you are working on things like maybe drug substances, those who do some extractions and they want to see the effects on maybe some culture or anything. You sometimes mistakenly you use this word synergistic effect, which actually is not what you mean to say. Synergistic effect is not the same as summation or additivity. These are two different things. But a lot of times the mistake is made that a summation effect is being referred to as a synergistic effect. So pay attention to this. Potentiation or synergism is the phenomenon of one substance that is having a certain effect, and a second substance which is not having any effect. But when you have the substance which is having no effect, you introduce that into the presence of the substance that is having an effect, it increases the effect of that substance. So let's use A and B, because we have here A and B. So let's say that you have substance A, which is having a therapeutic effect, and you have substance B, which is having no therapeutic effect. Now, even though substance B is having no therapeutic effect, when you introduce it in the presence of substance A, it increases the effect of substance A. So that's what you are seeing. The effect is increased. That's why we say it's potentiation. You are increasing the potency of substance A by introducing substance B, even though substance B is not having any effect. So you do have that, which is different from summation or additivity. Summation or additivity means that both substance A and substance B are having therapeutic effect. And when you put the two of them together, there is a combination of their therapeutic effects. Then we have what we refer to as antagonism, which is being referred to with the third curve, which is curved downwards. That is to say that if you have, so that's the opposite. Antagonism is the opposite of potentiation or synergism. To say that if you have substance A, which is having a therapeutic effect, you have substance B not having any therapeutic effect, but when you put the two of them together, it reduces the effect of substance A. Well, antagonism can also be as maybe the two of them may be having some effect, but when you put the two of them together, the effect is rather reduced. That is antagonism. In any case, it means that substance B, you can look at it as a substance B is antagonizing substance A, or substance A is antagonizing substance B, reducing the effect of one substance by the other. All right. Now, I think we can just move on now and quickly. We have done a lot. I mean, we talk about cell communication and talked a lot about cell receptor interaction, referring to receptors on the surface of the cell. We even refer to receptors that are inside the cell in the cytoplasm, internal receptors. So, I mean, much of this we already understand. And it is just that in place of, say, ligand, let's take the time we were looking at this, we looked at it when we were looking at the ligand as the hormone, and then a receptor that was accepting the hormone on the cell surface or inside of the cell. So in this case, you can put in place of the ligand a drug substance. So it's a similar thing, similar effect that the drug substances do have. So that if, for instance, you are having a drug substance which is a hormone analog, it's only coming in to be stimulating the receptor of a particular hormone. Now, what we can talk about here is the fact that, yes, we also even, the first point, we also mentioned it, that receptors are selective. So a particular receptor which is stimulated by a substance is not the same for a different substance. So receptors are selective. So every drug substance will have, its receptors. Now we want to add two other things, which is to say that you can have what we refer to as silent receptors and active receptors. So silent receptors are those receptors that even when you have a ligand interacting with those receptors, there will not be any effect. I mean, so for instance, if you go back to, I think when we were looking at hemoglobin breakdown metabolism, we saw, in the process of the breakdown of the heme, you saw that there was a binding of bilirubin with albumin. In this sense, you see albumin as a receptor in the bloodstream, but when bilirubin binds to it, it does not have any effect. Similarly, even when let's just go to say the previous lecture where we were referring to the fact that you have drug substances that bind to blood proteins, and in this sense, you will have these drug substances binding to these proteins in blood and not having any effect at all. There's no effect from that. That's a silent receptor. But active receptors are those that when you have a drug that is, binding onto them, it results into a certain effect, like there is a stimulation. That is an active receptor. So if you have some maybe adrenogenic receptors, for instance, that you have, I think we looked at an adrenogenic receptor where you have adrenaline or epinephrine, for instance, binding onto it. And then it is stimulating a certain reaction cascade. You saw that cyclic adenosine monophosphate cascade of reactions, which results into transcription and translation of a gene in the nucleus. That will be an active receptor. All right. Now, one of the major, the first attempt to quantify the receptor interaction with, drug substances was by Joseph Clark, who was a British pharmacologist who, was part of the medical corps of the British Royal Army, served in both the First and Second World War, and later, worked in Cape Town University in South Africa, but later went back to London, where he continued to work as a researcher and, wrote a lot of books on pharmacology, pharmaceutical chemistry, and things like that. He was the first who, tried to quantify drug response and drug receptor interactions, which is actually being, sometimes you see it being referred to as the receptor theory. What you see as the Clark's occupancy theory, sometimes you see it being referred to as the receptor theory. It's a major theory upon which we study and understand how receptors interacting with substances. And what it says is that the effect of any substance is proportional to the area of the receptors that are occupied by the molecules of the substance. So if you have some receptors, you have a cell, and that cell has receptors on it. Now, the effect of a substance on that cell is proportional to the amount of the receptors that will be occupied by molecules of the substance. So let's say you have like 10 receptors that are on that cell. If you have one molecule that is bound onto one of the receptors, it will have a certain effect. If you have two, there should be an increase. It's expected that you have an increase of the effect. If you have three, that increase should continue. That is the proportionality that is being referred to, that the effect of a substance is proportional to the area of the receptors that are occupied by these molecules. And what it also means is that in order for you to determine the maximum effect of any substance on a substrate, or say a cell that is binding onto the substance, when you are comparing two substances, it means that the substance that is going to have the less amount of molecules that is binding onto the receptor and causing either maybe a stimulation effect or rather disabling that target cell, that substance is actually more potent than the substance which you need a higher number of molecules to do the same thing. I mean, simply put, if you have two substances, A and B, and you take, say, substance A, you take a concentration of, say, 0.1 mole per liter, and it is having a certain effect. And in order to have that equal effect that substance A is having at a concentration of 0.1 mole per liter, if you take a second substance, say substance B, that you have to take a concentration of 0.3 mole per liter of it, then of course, substance A is more effective than substance B, because you need a lesser amount of substance A to have that effect that you want. That if you take substance B, you have to take more of it before you have that same effect. So that's all what the Clark's occupancy theory summarizes. All right, now let's take a look at the general processes or the mechanisms by which you can have substances sort of inducing their effect. Now, let's take for it, let's say we have a pharmaceutical agent, it's a drug that can do different things. Now, first, a pharmaceutical drug can either be acting on an invader. Let's say that's a bacterial cell, can be acting on an invader directly. And how will it act on the invader? Some pharmaceutical drugs will attempt to cause destruction in the membrane structure. So there's a destruction in membrane integrity. Of course, if you have a bacterial cell having a membrane or even a cell wall, and you have a drug substance that goes to disrupt the structure, obviously that bacteria is going to break down. That will be what maybe you refer to as a bactericidal effect, because the structure of the bacteria is disrupted. So its content will just break open, and everything there will no longer be that bacterial cell. So that will result into the cell death, the bacterial cell death. Another thing is that, so what you refer to as the bacteriostatic effect, where the integrity of the structure of the bacterial is maintained all right, but the pharmaceutical agent targets a particular metabolic process of the bacteria. The substance targets a particular metabolic process of the bacteria that it goes to inhibit it. So that when it inhibits it, then it sort of slows down or stops the growth of the bacteria. It doesn't result in the death of the bacterial cell, but the growth and even, proliferation, reproduction of the bacterial cells will be inhibited. So you can have that pharmaceutical agent acting on the invader in these two ways. Pharmaceutical agent can also act on, say, the human cell in different ways, even more different ways. First, we saw how the receptors were acting in terms of signaling. So if this is a receptor in a human cell, that receptor responds to a certain stimuli from outside, say maybe a hormone or anything. But this time around, it is the pharmaceutical agent that is interacting with this receptor. And when it interacts with the receptor, of course, that signal, that message that the pharmaceutical agent brings is then transferred. It means you can have that as what we saw with the second messengers, all the way sending the message into the nucleus, where there will be the expression of a particular gene to respond to whichever process that is supposed to be controlled. That is to say, the transcription and translation will produce a particular protein, and that protein is going to have a certain ultimate response in the end. So it is either you have the pharmaceutical agent either stimulating that receptor or blocking the receptor. That's why we have plus or minus. So you can have that happening to the cell. Another thing that the pharmaceutical receptor can do is we have membrane proteins that serve as transport proteins. So you have these transport proteins which help in the transportation of various metabolic precursors, which will get into the cell, that will be used for biosynthesis and the generation of energy. So if you have a pharmaceutical substance that comes to block that transport membrane, then of course it's going to block the transport of those metabolic precursors that are coming into the cell, which will mean that the metabolic processes which should occur will no longer be occurring. Or the pharmaceutical substance comes to inhibit enzymes, metabolic enzymes that are in the cell. These enzymes are supposed to catalyze the biochemical reactions, and the pharmaceutical agent comes to inhibit those enzymes. Then that, of course, is going to inhibit the whole of the metabolic process that is being targeted. Or we have what we refer to as some voltage-gated ion channels. Even not only voltage-gated, we have some which also refer to as ligand-gated ion channels. So the difference is that you have a ligand-gated means that you have a particular substance, let's say calcium, for instance, that comes to bind onto a particular channel, or let's say the drug substance that we are referring to is what comes to bind because it's a pharmaceutical agent. It binds to that, and then what happens is that it triggers a change in potential, because what happens is that the channel opens or closes depending on the changes of the potential that is generated. Because what happens is once the channel is opened, you have ions that are moving across the cell. And once you have these ions moving across the cell, it generates a potential across the cell, either plus or minus. So for instance, if you are having hydrogen ions that are moving out of the cell, it means you are having positive ions moving out of the cell. And if you don't have an opposite, movement to, compensate for that positive charges that are moving out of the cell, then in the end, what you have is that you have inside the cell being more negative than outside the cell. That will cause a certain, potential across the cell. You see this mostly in say in the central nervous system. You have it also with the cardiac tissues. You see some of this, that which is responsible for contraction, relaxation of the muscles, or signal transduction across cell membrane, the nerve cells across the synapses. All right. Now, with this overview of the various scenarios that pharmaceutical agent can take, what we'll do now is that we take some specific examples of some drug substances and how they act. So that's to say, we now will focus on mechanism of action. I mean, different ways. Of course, we cannot talk about mechanism of action of all drugs. That's just not possible. Even if you take the whole day, you can't do that. You have to now take all the lists and begin to classify these drugs before you begin to see how they act. And of course, some of them you can group them and then you talk about their mechanisms of action. Now, before we talk about mechanism of action of some antibiotics, we need to understand a process of the synthesis of what we refer to as tetrahydrofolate. In other words, folic acid. In the body, we synthesize folic acid. So apart from, say, folic acid, which is taken as a supplement, that is if it is needed when there is maybe deficiency or is needed in much more amounts, then some supplements are taken. But the body synthesizes folic acid. It's important for the proliferation of cells, cell growth, particularly in our developmental stages, when the human developmental stages, even in developmental stages of, let's say, even bacteria, prokaryotes, eukaryotes, in developmental stages, they require the folic acid for cell proliferation and growth. So we have, from the beginning, the top, we have a dihydrotyrene pyrophosphate. I mean the red compound. And you have 4-amino benzoic acid. That's a blue compound. These two conjugate. So the two of them, gets conjugated. It's just you have a removal of that diphosphate. The pyrophosphate is removed, and you see that there is a conjugation. This NH2 here, it loses one hydrogen. Of course, that will go onto the diphosphate there, and you just have an NH. So it's just that you have a joining of these two molecules, the 4-amino benzoic acid and the dihydrotyrene pyrophosphate. And that gives you a dihydroperoic acid. That's the first reaction. Then from there, what you see is that there is a conjugation of a third molecule here, and that molecule is glutamate. or glutamic acid. That's an amino acid. The next thing you see is just a reduction reaction, where you see that, this double bond here, this double bond is removed, so that in place of that, you now have a hydrogen on nitrogen, which means, of course, there will be a hydrogen which will also come on that carbon there, between which you are having the double bond with a nitrogen. All right. So this a simplified reaction pathway for the synthesis of tetrahydrofolate, or say folic acid, which consists of these major components: the terine nucleus, the aminobenzoic acid, and glutamic acid. Now I want you to pay attention to something. On the first arrow there's a small note, sulfonamides, and there's a cross around across that arrow, which is to say that sulfonamides they are inhibitors of this enzyme, dihydroterate synthase. This enzyme, which catalyzes that first reaction, sulfonamides, which are a group of antibiotics, inhibit this enzyme. There's another one here, the third arrow below here. There's also a small note there, trimethoprim. Trimethoprim is also an antibiotic, which also serves as an inhibitor to this enzyme, dihydrofolate reductase, which is responsible for catalyzing this reaction. It's a reduction reaction where you have a hydrogen added on the nitrogen and the carbon there, so that you have that double bond removed. All right, so now let's zoom in on those. Let's zoom in on those reactions. That first reaction there and that third reaction there, where you have these antibiotics acting. Now, what it means is that sulfonamides are inhibiting dihydroterate synthase. So the enzyme which is being inhibited is dihydroterate synthase. The substrate, upon which you have these sulfonamides designed, is 4-aminobenzoic acid. And that's to say, if you take a look at, say, this is a structure of sulfanilamide, one of the sulfonamides. The sulfonamides have such a structure. Now, when you compare it to the para-aminobenzoic acid is the same as 4-aminobenzoic acid. If you say para, it means opposite. So you have the amine group and the carboxylic acid group opposite each other. So that's why we say para-aminobenzoic acid. Or you can also say 4-aminobenzoic acid. That is, if you count that carbon on which the carboxyl group is as carbon 1, and you count clockwise, the carbon on which the amine group is carbon number 4. And if you take a look at the structure of sulfanilamide and para-aminobenzoic acid, you see there's a similarity. And the difference you see is that in place of the carboxylic acid group in para-aminobenzoic acid, you have this sulfo-amine group there. That's the difference. And so it's more like the substance deceives the enzyme dihydroterate synthase, to think that the antibiotic is that 4-aminobenzoic acid and binds to it. And once it binds to it, it inhibits its activity. It can no longer bind to the 4-aminobenzoic acid to have the synthesis of the folic acid initiating. So of course, if we are saying that the synthesis of the folic acid is significant, is important for cell proliferation, cell growth, which is also for the bacteria. Then, of course, when you have sulfanilamide going to, bind onto the bacterial dihydroteresynthase, it will prevent that process, the synthesis of the folic acid. And once it prevents the synthesis of the folic acid, it means that it will inhibit the growth and proliferation of the bacterial cells. Similarly, trimethoprim, which is also an antibiotic, I think is used much more in urinary tract infections and maybe some more serious infectious conditions. You also have it also inhibiting dihydrofolate reductase. So, this process where there's a reduction of the dihydrofolic acid to the tetrahydrofolic acid, you have trimethoprim inhibiting that enzyme there. And when you look at the structure of the trimethoprim, not so much like that. The first one we saw was more closer to it. But here you see there's a significant difference, because if you look at this ring here, these two rings are fused. But here you are having the two rings, having some CH2 between them. So they are far apart. They are not fused. And you also even have some other groups, methyl groups that are attached on that ring there. You don't have a third ring. So which is, this should tell you that trimethoprim will be more of a broad spectrum than being more specific, like, say, the sulfanilamide, because it's not very closely, an analog of the substrate. So even though if you can have the ring binding into the active center of the enzyme, dihydrofolate reductase, and inhibiting it, it means that it will not be as specific as another, drug which will, be much closer to that structure. So let's look at another one. Methotrexate is also one of these drugs which are used particularly in some think cancer, to prevent cell proliferation of cancer cells. Like in chemotherapy, you have this used. And if you take a look at the structure of methotrexate, you can see clearly that it looks very, very, very much like the substrate, dihydrofolic acid. Looks very much like it if you look at the structure. I mean, the differences you see is, positions of double bonds. For instance, if you look at the first ring, you can see that you are having, different positions. You have three double bonds here. Here you have only two. And if you look at the second ring, the same thing. So you are having, it's not the same molecule, but when you pay attention to it, you realize they are very much different. If you look at the first ring too, there's an amine group there, which is not there in the dihydrofolic acid. It's rather a double bond O. There's even a methyl group on this nitrogen in methotrexate. That methyl group is not there in dihydrofolic acid. So there are significant differences in the molecules, but the structures look much, much more similar, which means methotrexate will be very, very much more specific in its mechanism of action in the prevention of the proliferation of cancer cells. All right. Now, Of course, I know maybe you will start having some questions like, so if because you do have synthesis of um, the tetrahydrofolic acid also, as a normal process which is occurring in us. So if you are having some medicines like this, which are going to inhibit the synthesis of tetrahydrofolic acid in bacteria or say in cancer cells, how about the normal process? Won't it affect it? Yes, of course, it does have such side effects. That's why sometimes you have, say, chemotherapy, maybe resulting into things like hair loss, for instance, or scaly skins, because even though it's going to um, prevent proliferation of cancer cells, it does affect the normal process also to some extent. And because of that, it begins to affect the normal growth of, say, the skin and its appendages. All right. Now we take another one. But before then, we'll take a look at, let's zoom in on the structure of the cell wall of gram-positive bacteria. So below here, if you take this to be some bacteria, and we zoom in into the membrane and cell wall, what we see is, so the membrane that we know is a phospholipid bilayer, and the cell wall, which is the outer covering of that bacterial cell, is made up of what we refer to as peptidoglycan, which is a polysaccharide that is linked with some oligopeptides. So, I mean, in this structure, we have a key up here, the violet spheres or violet circles are N-acetylglucosamine residues, and the blue circles are N-acetylmoramic acid residues. Then you have some cross-linkage between them, and this cross-linkage is joining these oligosaccharides, because this N-acetylglucosamine, N-acetylmoramic acid, they are derivatives or modifications of monosaccharides. And you are having these peptides that are binding these oligosaccharides, in that manner. So you have that as some peptide, chains that are forming some cross linkages between these oligosaccharides. So it sort of forms a certain, well meshed together wall, strong, giving that protection to the bacterial cell wall. All right, having seen that structure of the bacteria cell wall, let's also take a look at the synthesis of that, how that happens. Because a particular enzyme that will be targeted by an antibiotic. And that enzyme is what, so if the drug is penicillin, and so that enzyme is referred to as a penicillin binding protein. That is, if you are referring to the drug that is binding onto the protein to name it. But actually, the process that the enzyme carries out is to form the cross linkages between the peptides that are on the oligosaccharides. So the enzyme is a glycopeptide transpeptidase, sort of transferring these peptides to form some linkages between them. Now, if you look at it from step 1 to step 5, what is happening is that in the active center of the enzyme, you have a reactive serine residue. The amino acid serine residue is there. And you see that this enzyme is having some two pockets, a longer pocket and a shorter pocket. No, look at it. It looks more like a chair, or say a small H. All right. Now what you see is that first, so we come to stage 2, where now you see that you have one of the peptides binding into the small groove of the enzyme. Now, what you notice is that in the structure of the cell wall, you have, so this teal color refers to N-acetylglucosamine. So the key is here, NAG, that is N-acetylglucosamine. The green is N-acetylmuramic acid, which is NAM. So you have that sort of alternating structure where you have N-acetylmuramic acid and acetylglucosamine joining together to give you that polysaccharide chain, or say an oligosaccharide chain. And then on the residue of the N-acetylmuramic acid, the green one, you have the peptides. So on one chain like this, you are having a glycopeptide, because this is an oligosaccharide which is having some little peptide chains on them. And on each of these peptide chains, you have the peptides made up of, the red refers to alanine, the amino acid alanine. Blue refers to glutamic acid or glutamate, and the green refers to lysine. So that you see, the sequence of the amino acids which are in these peptides. Now, two of these glycopeptides are joined, they are cross-linked by the activity of the penicillin binding protein, or the glycopeptide transpeptidase. So you see that these two are getting joined, and that activity is being carried out by the enzyme. Now, in the end, after they are joined, that is between the lysine residue and one of the alanine residues. The alanine residue is what ends there. You have the green one. So there's that cross-linkage between the lysine and the alanine. And then the enzyme leaves. So now you see there's that cross-linkage which is between the two glycopeptides. Same thing will happen to these ones too. I mean, these 2, 3 here, they too, you have the glycopeptide transpeptidase coming in to link them up, so that you have, every 2 of these glycopeptides getting linked like that. And then together they will form that layer of the cell wall. Now that we understand that process of the synthesis of that, glycopeptide, we can now look at how an antibiotic, penicillin, which targets that enzyme, it targets that protein. So if you see these structures that are like 9 or 6 orange over here is just to represent penicillin. And we have seen the process of the formation of the cross-linkage between the glycopeptides. Now, in the presence of the glycopeptide transpeptidase, when you have penicillin coming into contact, it opens up. So penicillin binds into the active center of the enzyme. So you see the penicillin is bound inside the active center of the enzyme. And once penicillin is bound in the active center of the enzyme that way, the enzyme is rendered inactive. It can no longer carry out its activity. And so if you look at the structure, if you look at the structure of the penicillin. So this is a penicillin structure. And if you look at the structure of these peptides that are having the cross linkages between them, looks much like it, except that you see that this portion is opened. But for penicillin, you see that it is closed. See, this ring is closed. But for the glycopeptide itself, because this place is opened. And that is why you see penicillin is closed from the beginning, but when it comes into contact with the enzyme, the glycoprotein transpeptidase, it opens up and then binds into it. Sort of sometimes referred to as a suicide, mechanism, sort of as if it's not active, but the moment it comes into contact with the enzyme, then it opens up and binds to it. All right, so there, yes, the enzyme which is being inhibited, glycopeptide transpeptidase, you have the substrate there being these peptidoglycans, the ends that are being targeted. And the inhibitor is the penicillin, which is doing that inhibition there. Now, you see this ring, you'll notice, we'll talk about that ring much later. You have that you can look at it much more this way, that if this is the enzyme, the glycopeptide transpeptidase with an active center having that serine residue there. So the serine, if you look at the structure of serine, the side chain of serine is CH2OH. And you see penicillin now binds onto that serine residue to form that penicillyl enzyme complex and renders the enzyme inactive. You can look at it, in a much simpler way, this way. Now, you see, I think if you look at, is it Tobinco or Entrance Pharmaceuticals and some other pharmaceutical companies in Ghana? Yes, they do produce some of these antibiotics that they refer to as beta-lactam antibiotics. And you see the ring structure, that functional group inside these antibiotics is what is being referred to as the beta-lactam rings. So you have this four-sided ring, which is having this ketone group there. That's a lactam. It's a lactam. And you have different types of it, so that you have this having a five-sided ring fused with sulfur there. But if you look at this and this as if they are the same, but they are not, because you have a double bond there, which is not in this. And when you come to the next ring, there's no sulfur. And when you come to the next, you see it's a six-sided ring with sulfur there. Even though this is also six-sided ring with sulfur, there's a double bond there. So you see these similar rings in the beta-lactam antibiotics, which act just like how, the penicillin acted, what we just looked at. So you have these different rings, like the first one we refer to as a pinam ring. The second is a pinim ring. You have a carbapenem ring, cfam ring, and then another cfm ring. Of course, this ring is not exhaustive. There are more rings that you can check. You see some more of these beta-lactam rings. And a common antibiotic on the Ghanaian market, amoxicillin. We do have amoxiclav, which is a combination of amoxicillin and clavulanic acid. And if you look in the structure of amoxicillin, you see the beta-lactam ring is there. That's the active part of the antibiotic that opens up and binds to the penicillin binding protein. Clavulanic acid also looks much okay. Even the ring of clavulanic acid is not present here. This is there's an oxygen here, which is not part of these rings. And this is an oxypenam ring. But the ring which you find here in amoxicillin, I think is the first one. So that is a penam. An amoxiclav that you find on the market, I think that's because, you know, the bacteria is able to develop resistance to these antibiotics by the production of the enzyme lactamase. So the lactamase does, break down these antibiotics. But you have the clavulanic acid that binds to that lactamase, deactivates it. So which is why you have that combination of amoxicillin and clavulanic acid as amoxiclav on the market. It's not sort of deal with that antibiotic resistance. All right. Now, to take a look at some, mechanism of killers, pain relievers. Let's take a look at the process of the biosynthesis of prostaglandins. Prostaglandins are some, let's say, natural pain relievers in the body. They deal with inflammation. The body synthesizes them. And you see it's starting from diacylglycerol of phospholipid. And you see, we've seen this, I think when we were looking at some mechanism of action of some hormones, we've seen diacylglycerol, phospholipid, we've seen those things. So you see phospholipase C, phospholipase A2, they act in a breakdown of these diacylglycerols to get arachidonic acid. And from this arachidonic acid, you have these cyclooxygenases. What is you see here? COX-1, COX-2. I think some of you who are working in some pharmaceutical, or maybe in the pharmacies or pharmaceutical companies, you may have come across drugs. I think there's a drug named COX-1, COX-2, things like that. And this is what is based on, because you have these cyclooxygenases which are involved in the action of these proteins from arachidonic acid, and these prostaglandins go into the form of thromboxanes, which act in blood clotting, or you have prostacyclins, which will do the opposite, rather, preventing blood clotting. I mean, together they act to relieve pain in the body. I mean, so what really, not to relieve pain, but to recognize pain. So these prostaglandins, what they do is for pain recognition, because wherever there's an inflammation, there will be some kind of clotting there, and that will be resulting into the kind of pain that you will be feeling in those places. And the process of the synthesis of these prostaglandins, you see these cyclooxygenases that are acting on arachidonic acid to give you, the structure of prostaglandins. Now, what you do see is that, a drug like brufen. So if you zoom in on those two first reactions, where you have, so first, let's just take the second reaction where you have arachidonic acid, where you have cyclooxygenases acting in the cyclization of arachidonic acid to form prostaglandins. You have brufen coming in as an inhibitor to those cyclooxygenases, which is to prevent the formation of these prostaglandins. And once you have this prostaglandin formation being inhibited, then of course the pain recognition will be reduced. So in effect, you have a reduction or you have a relief from the feeling of pain. All right. Now, similarly, you have another painkiller like aspirin also doing the same thing here too. Aspirin is inhibiting cyclooxygenase 1, which is involved in that process of cyclization of arachidonic acid into, prostaglandin H2. I mean, this is happening in platelets, where there is an inflammation. You have, a response to it by, platelets getting activated, and that's prostaglandin goes on to, formation of some thromboxanes, and from prostaglandin H2 to prostaglandin E. These are different forms of these prostaglandins, which of course will induce some signaling pathways. And then there will be some, it can even result into like, tumor growth, for instance. So because, clotting and you have different responses from the intestinal epithelial cells, for instance. So you have aspirin blocking that process, and then that will be resulting into some pain relief. Now, if this looks more like, will I say physical? Well, not physical. You can still say it's a chemical. So it's a mechanism of antagonism. By say we have these antidotes, say diethyl antidote, that is used for the treatment of intoxication from heavy metals. And heavy metals, when they get into the body, intoxication from heavy metals, they go to bind to proteins. It can be some enzymes, can be some hormones, it can be some neurotransmitters, which means that a heavy metal can be, by binding to some proteins which are maybe hormones, can be resulting into some hormonal imbalance, or maybe if they bind to some enzymes, they will be preventing some metabolic processes, for instance. So if you look at the structure of this diethyl antidote, you see the ends are some sulfur hydral groups, which looks very much like say, you have in proteins also, say, the amino acid cysteine, between which you may be having some bridge, some disulfide bridge. But when a heavy metal comes to bind onto those cysteine residues. But the diethyl antidote, looking very much like that particular part of the protein, can then come to bind to the heavy metal. So when it binds to the heavy metal, it then sets that protein free. So the protein can then be active. So it's one way of treating intoxication from heavy metals. And then you see that once the antidote binds onto that heavy metal, the heavy metal can be excreted out of the body in that antidote toxicant complex. Can be excreted through urine, for instance. The protein is set free, and there is normalization of metabolic processes. Now, I think we want to look at maybe the mechanism of action of some drug like maybe heparin, for instance, which is used to prevent coagulation. They say it's an anticoagulant. So how anticoagulants will act. To do that, we need to look at the process of coagulation. So we see platelets that, upon inflammation, you have some platelet activating factors. I think we did look at platelet activating factor as a lipid. I think when we were doing the hormones, we may have looked at it. And you see the platelets when activated. So these are platelets, and then these two just shows activated platelets. And these activated platelets go further to activate several factors. So these are proteins that they are activating. They have different proteins in different phases, initiation phase, and then amplification phase. You see that occurring. But don't bother yourself about this. All of these conversions in the initiation phase, amplification phase. Let's focus here. Where you have the factor XA. Let's say factor 10A. Factor 10A is activating prothrombin to thrombin. And that thrombin then converts fibrinogen, the protein fibrinogen, to fibrin. And it is that fibrin that forms a mesh, to trap blood cells, red blood cells. So this fibrin forms some cross linkage, and then once it traps, the blood cells, you then will have a clotting of the blood to prevent any further, bleeding. You also have, or maybe if it's an internal this into that, let's just say this would then result into clotting. And you can have the thrombin also going into the conversion of factor 13, to its active form. So factor 13 is inactive, and then it converts it to its active form, which will then also be acting directly on the stabilization of the fibrin. And now what we'll do is, so heparin actually will be acting on this. So pay attention to this, because I said we should come down here, forget about these things, and focus on this factor 10A, which is written as XA. So, and then on the conversion of this prothrombin to thrombin, coming down to the formation of the fibrin. And we see heparin. So heparin in different forms. We have, so they are all different, like just different forms of heparin that, bind to a protein, antithrombin. Antithrombin is in its inactive form, but when you have heparin coming to bind onto it, it activates it. And when it activates it, you have that activated antithrombin binding, onto the factor 10A, the factor XA. So you have factor XA, which is shown in red there, which is binding on that activated antithrombin. I mean, these structures up here are just referring to the, say, different forms of heparin, which is an anticoagulant. And it binds onto antithrombin, that is to activate it. And that activated form of the antithrombin binds onto the factor XA. And you saw the factor XA involved in the conversion of prothrombin to thrombin, which went down that pathway into the formation of the fibrin. And once you active antithrombin binding on XA, it then inhibits it. It's no longer able to result into that formation of the fibrin. That will result into the anticoagulation effects. Prevention of what is being referred to as thrombosis, coagulation, or say formation of blood clots in a blood vessel. All right. I think maybe this should be the last one, which is alcohol dehydrogenase antagonism by ethanol. Is another mechanism that is a common one, that methanol intoxication. When persons are intoxicated with say methanol, which is toxic, which can result into the formation of formaldehyde and formic acid, causing very serious metabolic acidosis, can result into blindness. It can result into nervous system breakdown, even fatality. But because the alcohol dehydrogenase also acts on ethanol, when in such a methanol intoxication, alcohol is introduced into the body. Ethanol is introduced into the body. It goes to bind onto the alcohol dehydrogenase. And when it binds to the alcohol dehydrogenase, it rather produces less toxic metabolites. And because in the human system, you have the alcohol dehydrogenase being able to convert ethanol to acetaldehyde, which then gets converted to acetic acid. That acetic acid, can easily bind onto a coenzyme A, form acetyl-CoA, and then goes into the TCA cycle, generating energy. So those metabolites, acetaldehyde, and then quickly gets into acetic acid, those are less toxic metabolites than the metabolites formaldehyde and formic acid. In that way, the intoxication of methanol is treated in that manner. All right. Okay, maybe this should be the last. What time is it now? Okay, I think we still have like some 25 minutes, so it's okay. So we have what you also refer to as say beta blockers. If you look at the right hand side, this is a mechanism which we looked at when we were looking at the mechanism of action of the hormones. So you see here, adrenaline, noradrenaline. So we actually looked at this mechanism of action where you have adrenaline or noradrenaline binding onto receptors, membrane receptors that are bound to some what you say G proteins. And you saw there was that activity of phospholipase C, which resulted into the formation of the phosphoinositol or inositol triphosphate. And we saw that reactions into the formation of diacylglycerol, calcium ions going into the activation of other proteins into some biochemical responses. You find this happening, it can result into some smooth muscle contraction. So in the heart, you do have say, let's say adrenaline and noradrenaline will be acting on what we refer to as beta receptors. So this particularly, so you see, if you look at these receptors, you see we label them alpha 1, alpha 2, and this is the beta receptor. So let's focus on this last one. So this beta receptor, when you have the adrenaline or noradrenaline acting on it, we have seen this reaction where there's the conversion of ATP to cyclic adenosine monophosphate, which is a second messenger. And that second messenger will be involved in a cascade of reactions, which then will result into cardiac muscle contraction and relaxation, which is, responsible for, the rhythm of the heart, or let's just say relaxation and contraction of the muscle, or say blood pressure, for instance. Of course, this also acts in the process of glycogenolysis because it's supposed to promote the breakdown of glycogen in the liver to generate energy, because that's what adrenaline does. Adrenaline and noradrenaline, they promote the process of glycogenolysis. All right, so if that's understood, we see that you have so it's a peripheral nerve that is releasing adrenaline. So this is just to show like a synapse where you have adrenaline being released, and that adrenaline is supposed to bind onto the beta receptor, which is what we are showing at the right-hand side here. The beta receptor is embedded in the membrane of the cardiac cell, but you have beta blockers that when they bind onto this beta receptor, they then prevent stimulation from, let's say, adrenaline on the beta receptor. And so there will be no action in the cardiac cell. That way, you have the beta blockers, reducing blood pressure in cases of persons having high BP, where there is that high contraction and relaxation of the muscles. You have some of these drugs which are used in cardiac conditions as beta blockers. All right, I think I've talked enough. It's some 20 minutes or so to 2 hours. So I will stop here and open the floor. If you have anything, you can write in the chat or you can raise your hand. I will enable your microphone and then you can unmute yourself and speak. The floor is open.